Dear students, today we are going to discuss on the topic mitochondrial electron transport chain. This is a very important and interesting topic to be discussed. What is the definition of mitochondrial electron transport chain? This is the final common pathway in aerobic cells by which electrons derived from various substrates are transferred to oxygen. Electron transport chain, shortly called as ETC, is a series of highly organized oxidation reduction enzymes whose reactions can be represented as reduced A plus oxidized B gives oxidized A plus reduced B. This electron transport chain is localized in mitochondria. The outer membrane of mitochondria is permeable to most of the small molecules. The inner membrane shows a highly selective permeability. It has transport systems only for specific substances such as ATP, ADP, pyruvate, succinate, alpha-ketoglutarate, malate, citrate, etc. The enzymes of the electron transport chain are embedded in the inner mitochondrial membrane in association with the enzymes of oxidative phosphorylation. The most accepted sequence of electron carriers in mitochondria is the first electron donor will be the substrate. The substrate is going to be oxidized and the electrons or hydrogen equivalents are first given to NAD. NAD is therefore reduced to NADH and H+. And the reduced NADH is reoxidized in electron transport chain and giving back reducing equivalents to FAD. FAD gets reduced to FADH2 and the reduced form of FADH2 in ETC is reoxidized, giving those hydrogen equivalents to CoQ or ubiquinone. This CoQ gets reduced and this reduced CoQ gets oxidized in electron transport chain and gives its reducing equivalents to cytochrome B. From cytochrome B, the electrons are transferred to cytochrome C1, cytochrome C and cytochrome A3 complex, and finally the reducing equivalents are given to half molecule of oxygen to form a molecule of water. The redox potentials in the electron transport chain are in increasing order except in the case of ubiquinone. The chain actually consists of a series of redox couples. At each step, electrons flow from the reductant of a redox couple with more negative redox potential to the oxidant of the next redox couple having a more positive redox potential. Dehydrogenation of substrate is the first step in the process of respiratory chain oxidation. Most of the dehydrogenases require NAD, which can accept a hydride ion which is formed by one hydrogen atom and an electron. The electron is received from the second hydrogen atom, releasing the second hydrogen atom in the form of a proton called H+. The second type of dehydrogenase reaction makes use of FAD as a coenzyme. In these reactions, the oxidants are reduced to NADH plus H plus or FADH2. The electron transport chain in the mitochondrial membrane has been separated into 4 complexes. They are complex 1, complex 2, complex 3 and complex 4. Complex 1 is otherwise called as NADH CoQ reductase. Complex 2 is otherwise called as succinate CoQ reductase. Complex 3 or CoQ cytochrome C called as cytochrome C oxidase. Complex 1 or NADH CoQ reductase has two important functions. One is electron transfer and other one it acts as a proton pump. The system catalyzes transfer of two electrons from NADH to small lipid soluble CoQ via FMN and FES clusters. From FMNH2, electrons are transferred to a group of FES proteins. The electrons are then transferred to CoQ. The process is accompanied by pumping of protons from mitochondrial matrix into intermembrane space, and this complex permits 1 ATP formation at site 1. Next is complex 2 or succinate CoQ reductase. Flow of electrons from succinate to CoQ occurs via FADH2. Succinate plus CoQ gives fumarate plus CoQH2. The small energy change does not allow succinate CoQ reductase system to pump protons across a mitochondrial membrane. Hence, this protein complex does not contribute to proton gradient. Hence, no ATP is formed in complex 2. CoQ is the electron acceptor in the reaction catalyzed by NADH CoQ reductase, that is complex 1, and succinate CoQ reductase, that is complex 2. The electrons received are subsequently transferred from CoQH2 to CoQ cytochrome C reductase, that is complex 3. Complex 3 or CoQ cytochrome reductase has two important functions. The first function is it acts as a proton pump, and the second function is it catalyzes transfer of electrons. This system catalyzes transfer of electrons from CoQH2 to cytochrome C via cytochrome B and cytochrome C1. Fe3+ accepts electron and is oxidized to Fe2+. The system also acts as a proton pump like complex 1. It is believed that 4 protons are pumped across a mitochondrial membrane during the oxidation. The energy change permits ATP formation, which is called as site 2. The next complex is complex 4, otherwise called as cytochrome C oxidase, which has two important functions. One is it acts as a proton pump, and other one it catalyzes transfer of electrons to molecular oxygen to form water. This is the terminal component of ETC. It catalyzes the transfer of electrons from cytochrome C to molecular oxygen via cytochrome A, copper ions, and cytochrome A3. Copper atom adjacent to cytochrome heme A is CuA, that is copper A, and cytochrome heme A3 is close to copper B. From cytochrome C, the electrons are transferred to heme A CuA cluster and then heme A3 CuB cluster. The system also acts as a proton pump. It pumps 2 protons into intermembrane space. The energy change permits ATP formation between cytochrome A3 and molecular oxygen. At 3 sites, free energy released per electron pair transferred is sufficient to support the phosphorylation of ADP to ATP, which requires about 8 kilocalories per mole. Now we are going to discuss on the topic ADP is to O or P is to O ratio. What is this ratio? It is a measure of how many moles of ATP are formed from ADP by phosphorylation per gram atom of oxygen used. This is usually measured as the number of moles of ADP that disappear per gram atom of oxygen used. P is to O ratio is equal to phosphate group esterified divided by electron pairs transferred. Inhibitors of electron transport chain. Transfer of electrons is selectively inhibited at various components of the electron transport chain by a variety of substances. Some of these are used as poisons and some which are used as drugs. The inhibitors of site 1 at complex 1 are rotenone, amobarbital, pyridine A and certain drugs. Rotenone is a fish poison and also an insecticide which inhibits transfer of electrons through complex 1 NADH CoQ reductase. Amobarbital and secobarbital inhibits electron transfer through NADH CoQ reductase. Pyridine A is an antibiotic which blocks electron transfer by competing with CoQ. Drugs, we can say chlorpromazine as an example of inhibitor of electron transport chain at site 1. There are some inhibitors which inhibit the ATP formation at site 2 with complex 3. They are antimycin A, dimercaprol and certain hypoglycemic drugs. All these inhibitors block electron transfer from cytochrome B to C1. And there are some inhibitors which inhibit at site 3 in complex 4. They are cyanide, hydrogen sulfide, azide, carbon monoxide. Cyanide, hydrogen sulfide and azide inhibit terminal transfer of electrons to molecular oxygen, and carbon monoxide inhibits cytochrome oxidase by combining with oxygen binding site. It can be reversed by illumination with light. There are some inhibitors which also inhibits complex 2. They are carboxin and TTFA. These two inhibitors specifically inhibit transfer of reducing equivalents from succinate dehydrogenase, and malonate is a competitive inhibitor of succinate dehydrogenase.